All right. Well, thank you everyone for making it back after lunch. I know it's been a long day and we've thrown a lot of new concepts and ideas at you. So I want to just describe to you guys. So I think one of the most exciting things about metabolic modeling is that not only can we predict fluxes, but we can also use measurements to calculate them. And that is that's what we're going to talk about today, is how to actually calculate fluxes from metabolomics measurements. So let's just talk about some of the goals for this tutorial today. So just to give you a little background here, in a 13C metabolic FluxOmics experiment, an organism is fed a substrate with a mixture of isotopic labels where the location of each labeled atom is known. But to fully specify the isotopic labeling state of the mixture, both the percentage of each component of the mixture and which atoms are labeled in each component must be described. So for today's FluxOmics tutorial, we're going to assume that all samples are taken when the organism is in both metabolic steady state, where the concentration of each metabolite does not change, and also isotopic stationary state, where the isotopic labeling state of each metabolite does not change. So if these assumptions are, even if these assumptions are met, hold on for just a second. Yeah, so even if these assumptions are met, mass spectrometry still cannot determine the isotopic labeling state of each metabolite that it measures. The best that it can do is measure the mass isotoper distribution, or MID. That's what this is right here, which represents a substrate with a mixture of labels where the location of each labeled atom is not known. So for a molecule with n atoms, this M0, M1, M2, M3 notation specifies the percentage of atoms that are either unlabeled or one labeled all the way up to fully labeled. So with that as the background, metabolic flux analysis takes as input the isotopic labeling state of the substrate, the MID of a set of measured metabolites, the metabolic model of the organism, like this, knowledge of the atom transitions, which I'll talk about in a little bit, and a known uptake rate, in this case, V equals 100, V1 here. And it uses that information to calculate the energy and material flows through the organism. So this is where the rubber meets the road, essentially. So the goal of today's tutorial is to understand how this calculation is done. We'll introduce some new concepts that build upon what we've learned this morning about flux balance analysis and elementary modes. And then we'll use a simple model to understand step by step how steady state fluxes can be used to calculate these mass isotoper distributions. And then, but that's not the interesting question, because if we had the fluxes, then we wouldn't need to measure the MIDs, but all we have are the MIDs, and we want to know what the fluxes were that produced them. So we also are going to use to understand how measurements of these stationary state MIDs can be used to solve for steady state fluxes. We'll also try to understand why the choice of tracers is critical for the ability to identify flux distributions, and also how to distinguish good tracers from bad ones. Okay, so any questions so far? Are we good?
We're good.
We're good, okay. So let's start off with a simple metabolic model. So in this system, metabolite A is the substrate, metabolite E is the product, that's the thing that we care about, and the intermediate metabolisms B, C, and D are assumed to be at metabolic steady state. So these guys aren't changing, and isotopic stationary state. So their MIDs are not changing. So you can have the same concentration of metabolite, but the MIDs might be changing over time as the tracer kind of flows through the system. So we're assuming these are not changing. The MIDs are not changing. This network has two free fluxes, namely the substrate consumption rate V1, that's what we measure. And then the flux of the reversible cleavage condensation reaction V4. So that's this guy right here. And this is the thing that we can't measure, but we'd like to be able to calculate. So we'd like to determine flux V4 based solely on the measurement of the MID of E and a known uptake rate of A. So that's V4 here. Okay, so and then one additional piece of information that we have is that we know which of these atoms in B go to which of these atoms in C and D, right? So this A reaction right here says ABC goes to ABC. So 1, 2, 3. So these metabolites right here go to these metabolites. These atoms here go to these atoms here. But for B going to and for B going to E, also it's just 1, 2, 3 goes to 1, 2, 3. That's straightforward. But this last one's a little trickier. It says B goes to C plus D, but in particular, the C metabolite, so number 3 here goes to C, and 1 and 2 go to D. Okay, and that's going to, so that additional atom mapping, these additional atom transitions are really what's critical in being able to recover what the flux of V4 is going to be. Okay, and just like beforehand, we had we have code that we can actually run. Now, unfortunately, I just didn't have time to get a binder up and running, but the code is available online. And I have a link for it, so you can install it locally. But I don't have a binder for you, so you'll have to install the software. And unfortunately, it relies on a couple of things, so you may not be able to get it to work. But just go ahead and follow along, and then I will after this tutorial is over, I'll go ahead and turn it into a binder so that everything is there for you to install. So we already have a question. As B goes to C plus D, what happens to type 3? I'm assuming counts of types 1, 2, and 3 are conserved. So I just want to make sure I understand your nomenclature here. So when you say, so what happens to type 3? So, oh, I see. So this guy right here goes back into it. So it's like it goes out and it comes right back in. It doesn't escape out of the system. Later on, I show an extended model where there is escape. But at least for this very, very simple model, we just have, as you know, B goes to C plus D, and then C plus D goes right back into B and then down to E. Did that answer your question, Arvin? Yes, okay, cool. Okay, so now we have a computational model, and we're now capturing these carbon transitions here. And when I click on it, then we get the stoichiometric matrix of the network, just like we had before. And this is what it looks like when we know what the fluxes are. So there's 100 going down this way, and there's 40 going out and 40 going back. And just like beforehand, so people asked in the last tutorial, can I calculate these at elementary modes? And the answer is yes. There's a special package called, what is it called? PyEFM. And I can put a link out to that in the chat window later. PyEFM allows you to calculate these elementary modes directly. We're going to be using this a lot in this tutorial. So there we go. So here are the elementary modes for this very simple model, and we can actually, oh, there you go, Peter St. John. Thank you, Junhin. And then we can visualize those elementary modes separately. And so what you see here is that there's 2 independent paths to producing E. First one, actually, there's just not even to producing E. So there's 2 independent steady states in this metabolic network. First one goes from A to B to E, and the second one just goes from B to C and D, and then back to B, and they just kind of cycles and loops like that. And so, but the thing is at B, things start to mix. And so if I have an initial metabolite tracer here, then when it goes here, it gets scrambled, and when it comes back, that scrambling gives you a clue as to what's happening. It gives you a clue as to how much flux must have been coming through here relative to this flux going here. And that's going to be the key to being able to disentangle this. Okay, so let's go ahead and introduce some new concepts. Now, the first concept is that of an elementary metabolite unit. And this is not an elementary mode. That's a completely separate concept. This is just, other than the fact that it's elementary, there's nothing else that's similar. Elementary metabolite units are distinct subsets of the compound's atoms. So for the 3-atom metabolite A, shown in the figure below, we have 7 EMUs. And so this is the first one. This is the shaded region shows you what the EMU for, so these are 7 EMUs here. And the first EMU is A1, and that just refers to the first atom of A. A2 likewise to the second atom, A3 to the third atom. But since these are all subsets, you can also consider all pairwise subsets. So A12 is the first 2 atoms, A13 is the first and third, and so on. And lastly, you have the subset of the entire set of metabolites, so A1, 2, 3. Okay. So these are the things that we're going to be playing with, and these are now going to be, these elementary metabolite units are now going to be our nodes in our graph rather than whole molecules. So next, we also need to introduce the concept of an EMU reaction. So this figure below shows 3 types of EMU reactions that we can distinguish. And we can talk about a condensation reaction where A1, 2, and B1 are elementary metabolite units, and they combine to form C1, 2, 3. We also have a cleavage reaction where it's actually A1, 2, 3 is the elementary metabolite unit. There's a 4th one here, but that gets cleaved off. And so you're left with C1, 2, 3. And then you have a unimolecular reaction where A1, 2, 3 goes straight to C1, 2, 3. So for each reaction type, we would like to determine the minimum amount of information that's needed to determine the mass isotoper distribution of the product C. So EMU C1, 2, 3. For the cleavage and unimolecular reactions, the MID of C1, 2, 3 is equal to the MID of A1, 2, 3. So note that for the cleavage reaction, the atoms of A that are not transferred to C1, 2, 3 are not considered in the EMU reaction. So their labeling does not affect the labeling state of C. For the condensation reaction, the MID of C1, 2, 3 is obtained from the convolution of the 2 MIDs, of the MIDs of these 2 EMUs, A1, 2, and B1. So let me just show you what a convolution looks like in this slide right here. So here's the MID of B1. Notice that B1 is could be unlabeled or fully labeled, and in this case, it's 100% fully labeled. And here's A12, and in this case, A12 is 0% unlabeled, 50% 1 labeled, and 50% 2 labeled. Okay, and we want to combine these 2 MIDs to produce the MID of C1, 2, 3. And the question is, how does this work? So first thing we do in a convolution is we flip B1 upside down. So it was like this, and now we're just flipping it upside down. And then we're going to slide B1 all the way down through A1, 2, and every time there's an intersection, right, blue and yellow makes green, then we perform a calculation. So when it's M0 and M0, then oops, sorry, I don't know how that happened. So the product of the 2 slots that were there, so this is M0 is here at 0, M0 here is 0. So you multiply 0 times 0, and then you put the answer in the M+0 slot of C1, 2, 3. And you slide it down one more, and now we multiply 0 times 1 and 0.5 times 0, and we take the sum of those and we put that in slot M+1. And we slide it down one more time, and this time M+1 times M+1 is 0.5 times 1, so that's 0.5, and 0.5 times 0, so that's 0.5. You add these 2 guys up together. And then the last one is 0.5 times 1, and that goes in the M+3 slot. So the result is that when you convolve B1 with A12, what you end up getting is this MID right here for C123. Does that make sense? Questions? Oops.
We're good.
We're still good. Okay, excellent. Could I repeat that? Yes. So I don't know if you guys have heard of convolutional neural networks. They work exactly the same way. The idea is that I'm going to take the MID of B1 and I'm going to convolve it with the MID of A12. And the way to do that is to flip B1 and then slide it down over A12, and everywhere they overlap, we're going to multiply the two together. So in this first case, we've slid down B1 so that it overlaps at the M plus 0 and M plus 0 are overlapping. And so 0 plus 0 is 0, so we put it in the M plus 0 slot, and 0 times 0 is the value that we put in that slot. Then we slide it down one more time, and we have now M plus 0, M plus 1 is multiplied. So M plus 0 of A12 is multiplying by M plus 1 of B1, and that number is still 0. And we add that to the number that we calculate here by multiplying the M plus 1 value with the M plus 0 value of B1. And again, that's still 0. And then we slide it down again. And this time, 0.5 times 1 is 1. So that's why this is bolded right here. If you take a look at 1 plus 1 is 2. And also 2 plus 0 is 2. So they all end up in the M plus 2 slot. And when you add these two together, you still get 0.5. And you slide down one more time. M plus 2 is 0.5. The M plus 1 of B1 is 1. So you multiply those two together, you get 0.5. And since M plus 2 plus M plus 1 is M plus 3, you put it in the M plus 3 slot. So now that is the result of convolving B1 with A12. That makes sense? Okay. Don't stop sharing. Just hopefully, there we go. I see what's going on here. Okay, cool. So that is how I take the MIDs associated with A12 and the MIDs associated with B1, and I compute the MID associated with C123. Okay. So, so that once you understand that little bit of trickery, the next thing to do is to combine EMU reactions into an EMU network. And what's cool about these EMU networks is that they decompose according to the size of the EMUs. So as I mentioned beforehand, we can have EMUs of size 1, size 2, or size 3 for a 3-atom molecule. And I can take all of the size 1 EMUs and build a network from them. Then I can take all the EMUs of size 2 and build a network from them. And then I can take all the EMUs that have a size 3, all the size 3 EMUs, and build one from them. Now, there's a slight caveat here, which is that this is so C1 plus D2 is composed of 2 EMUs. One is C1, the other one is D12. But when I convolve these two together, I get an MID that is of size 3. So it still fits in this EMU network size 3. Do we have access to the Jupyter notebook? Yes. So thank you, Avi, for clicking, for sharing that link. Okay. So now that we understand EMUs, EMU reactions, and EMU networks. Oh, sorry, there's one last step, which is I can separate these things out into separate networks, or I could combine them into one EMU model. And so the idea here is, So A123, B123, E123 fits right here. Then A3 joins with B3 and C1. Okay, so how does this work? So I start with A12 and that goes to B12, that's fine. B12 goes to D12, that's fine. And then I combine A3 with D12 to form B123. Okay. So we can also combine these EMUs into one giant network, where each of these are inputs to the network. A123, A3, and A12 are all EMUs where we actually know the isotopic labeling state for. So based on this information, then we can calculate E123. But we're going to stick with at least initially, we're going to stick with these EMU size specific networks, because they're going to help us calculate the MIDs of all of the EMUs. So how does that work? So we first solve for the MIDs of all EMUs of size 1 from the steady state fluxes, given the isotopic labeling state of the substrate. So let's just go ahead and take a look at what EMU network size 1 looks like. There are three EMUs, and actually only 3 reactions. These B1s don't really count. And this is what it looks like. So you have A3 going to B3, and then B3 going to C1, C1 going back to B3. And these fluxes are determined by the fluxes from the original diagram. So these fluxes right here, V100, V2 equals 100, V3 equals 40, and V4 equals 40. These are going, I'm going to go ahead and keep that around just so you guys can keep referring to it even as I'm going through this notebook. So let me go ahead and do that really quickly. So I think this is what, figure 1? Figure 1? Figure 1, yeah. Okay. So I'm going to keep this guy right here. And I'm also going to keep the EMU around. It could be helpful. Network 1 through 3. There we go. All right. So these guys are going to be here to refer to even as I'm going through this, okay? Do you guys want the elementary metabolite units as well, or are you okay without that? You don't know. Okay. All right. So now let's go back to our original experimental design. We have an isotopic labeling state. And from that isotopic labeling state, we calculate these MIDs. And the question is, how do we do that? So the answer is we start with a network size 1. And then what we do is we say, all right, let's take a look at these EMU balances. We know that A3 times V1 is equal to, so we say, so here's B3 here, right? So V1 plus V4 produces B3, and V1 times A3 plus V4 times C1 balances out, right? So all the fluxes going into B3 has got to equal the fluxes that were producing B3. Does that make sense? So can you see how these fluxes, this equation came from this network? Can I get a yay or an A, or do you want me to explain it in a different way? We're good? Oh, my music guy is typing. Good. Okay. All right. So we're good here. So we can see how we get this balance equation here, and we can see how we get for D for, is this right? This should be C3. C3. C1 actually. So then for C1, there's a reaction that produces, so B3, the B3 times V3 should be equal to C1 times V3. And that's what this shows right here. So now what we're going to do is we're going to rearrange these so that all the things that we're trying to solve for, B3 and C1, are on one side of the equation. And A3, which is the one thing that we know, we've got a measurement for that, which we can calculate right here. So it looks like A3 is 50% unlabeled and 50% one labeled. That's what we show right here. The A3 is 50% zero labeled and 50% one labeled. So we're going to take these numbers right here and we're going to solve for B3. So we say, so we just plug in the flux values. We know that the V1 is 100 and V4 is 40, and that's what this shows right here. And likewise, minus V4 minus V3, V3, so these are all 40, 40, negative 40. And we're going to multiply those by this flux V1 here, which is 100. We're going to invert this matrix, so that's where it goes right here, times V1, which is 100, times A3, and as I just showed here, the MID for A3 is 0.5. And so when you do that calculation, you end up getting, for B3, the EMU, the MID is 0.5, and for C1, it's 0.5. And so this is what, if we were to measure the EMU, the MIDs for these two EMUs, this is what they would look like. Okay, so now that we have the solutions to B3 and C1, let's go take a look at EMU network size 2. And so this is what that network looks like as well. It's exactly the same structure. Should it be C1 times V4? I'm guessing the answer is yes. C1 times, here we go, there's C1 times V, no. So it's only the arrows pointing to C1. So it's C1 times V3. And then you're balancing that with the thing that's pointing to C1. So that's B3 here. So B3 times C3 times V3. So the thing on the left-hand side, all the fluxes are pointing to it. And on the thing on the right-hand side, all the fluxes are pointing away from it. Does that make sense? Cool. So that was actually a clearer way of saying that. So these are the MIDs associated with B3 and C1 EMUs. Okay. So now let's go on to network size 2. And notice it's the same structure, but now we have different EMUs. So these are EMUs of size 2, but it's the exact same structure, so the calculation will be exactly the same. So in this case, the isotopic labeling state of A12 is 0.5.5. And the size balance says, okay, we just take B12, the arrows pointing to it are V1 and V4, and the arrows pointing away, pointing towards B12 from A12 is V1, and the arrow pointing to B12 from D12 is V4. So these numbers have to be, so these should balance. And likewise with D12, there's just V3 times D12 on the left side and B12 times V3 on the right side. And these all have to balance. So just rearranging terms so that B12 and D12 are all on the left side, and A12, which we know is on the right side. We can solve for B3 and C1. And here we have the solution for B12 and D12, and this is what their MIDs look like. Okay. Now let's see what happens when we go to size 3. So size 3 looks a little different. We got E123 and A123, and D12 and C1 pointing into B123. So that's what this guy is right here. And when we take this, we can, so our isotopic labeling state, now we have 2 isotopic labeling states, right? So you have A123, which was given to us. We have C1, which was calculated from the size 1 EMU network. And we have D12, which was calculated from the size 2 EMU network. And what we do is we take each of their MIDs, we convolve them together, and that is what this value is right here. And just to show how that actually works one more time, let me go ahead and do this for open evolution. So this is C1 times D12. So there's C1, there's D12, and what we're going to make is C1 cross or convolved with D12. So we take our C12, which is 0.5, and our D12, which is 0.5. We flip C1, and then we multiply C1 times D12 in the M plus 0 position, and we get 0 times 0.5. Then we take C1 in the M0 position times D12 in the M plus 1 position. That's 0.5 times 0.5, or 0.25. And this is 0, so when you add that, it's still 0.25. When you flip down again, then M plus 1 position times M plus 1 position is 0.5, 0.5 is 0.25. And then this is also 0.25, so you end up getting 0.5 total. And then lastly, 0.5 times 0.5 is just 0.25. And now we have calculated C1 convolved with D12. Okay. And hiding my floating meeting controls. Okay. So now that's how this 0.25, 0.5, 0.25 came about. So again, we do the same thing we did beforehand. B123, there's A123 going inside it from V1. V4 has the C12 across D12, and that's equal to V1 plus V4 B123. And likewise, E123 is pointing to it is pointed to by V2, and B123 points to E123 through V2. So we rearrange terms. We take the we plug in the values for the fluxes, and then we invert that matrix. We multiply things together, and we get a new set of MIDs. And this is what these look like. Okay. So how are people doing right now? 50%, under 100, you know, like where are we? Like, did I lose anyone? Is there something I could explain that would help make it more clear? Or has everyone just gone home? Makes sense. Excellent. Seems too much back and forth. Okay, fair enough. Okay, so 50%. Okay, fair enough. Is there, what would help to gain the other 50%? Okay, sorry. How are M0, M+1, M+2 calculated for A? Sorry if I missed it. Oh, so you didn't miss it. What happened is A was given to us. So that's, if we don't know the if we don't know the initial isotopic labeling state of A, then we can't do any of this, right? So the idea is that if we go back here to our original experimental design, A123 is given to us. We say we're going to choose the isotopic labeling state such that 50% of it is 1 labeled and 50% of it is 1, 2, 3 labeled. And from A123, we can then easily calculate what A1 is. So A1 is fully labeled, so its MID would be 0, 1. A2's labeling state would just be 50% unlabeled, 50% labeled, so its MID would be 0.5, 0.5. And A3's would also be 0.5, 0.5. And then I think we have another, I think we also have A12 somewhere. And so A12 would be 50% unlabeled, 50% two labeled. Sorry. What is A12? I forget. A12 is 0.5, 1 labeled, 0.5, 2 labeled. Here we go. Why do we keep 0 for the first position to D12? Let's take a look at D12 here. So when we calculate, wait, hold on. This is wrong. This should be B12 and D12. There we go. So why is D12 0 here is the question, right? And the reason why is because if you take a look at the isotopic labeling state, so the 1 position is always labeled. Second position might be labeled or might not be labeled. So D12 is, so here's D12. So A comes in, and then the 1, 2 part goes to D1, it goes to D here. And since it's always going to be at least 1 labeled, maybe 2 labeled, then we never have an unlabeled D12. Does that make sense? Okay, fair enough. But we okay, so that's kind of the answer is that because of the isotopic labeling state that we gave it, like the of A, does that mean that the A is the substrate that was given the 2 labels? Yes, 1 labeled at 1 carbon, the other labeled at 2 carbons. That's correct. Yes, that's right. So A is a mixture of 2 labeling states. We could have given it a pure tracer, and that this probably would have been a little easier to calculate. But it turns out, as we're going to discover next, that when you only introduce a pure isotope label, you cannot identify V4. And so we're going to explain why that's the case next after these initial round of questions have been addressed.
Another question, Discord. Do we do this for the entire GSM, genome scale model?
Oh, okay. Well, so yeah, so the answer is that, so first of all, there's, as you might guess from the fact that even with just a 3-unit, a 3-atom molecule, you get 7 elementary metabolite units, things blow up pretty quickly. But it's, you save a lot, right? So this is still better than the old way of performing flux balance analysis, which was, or metabolic flux analysis, which was with isotopologs or things. I mean, this is still exponential, but it's a much slower exponential than previous ways of doing things. So it is actually possible, and it has been done, to do 13C MFA at genome scale. Typically, they're done more at the central carbon metabolism, just because, you know, that's there are challenges in getting it to go genome scale, but some of the limitations have been lifted. So one thing is that we can easily calculate the atom mappings for every single reaction in a genome scale model. And the second thing is that there's been work by Hector Garcia Martin, for example, and also Costas Moranis on like efficient ways of, or constraining efficient ways to constrain these calculations so that they don't blow up when you go up to genome scale. But in general, yes, the way that I've described right now, if you were to do it exactly the way that I've described it, it would not work at the genome scale because there's just, you know, too many EMUs basically. So this is generally limited to kind of central metabolism without kind of adding a bit more sophistication. Okay. Any other additional questions?
Related to the labeled substrate.
Yep.
There was comment that I imagine that when supplying a mixture of differently labeled substrates, we should supply them as a 1-to-1 ratio.
It turns out the answer to that is no. Anything but 1-to-1 ratio should be fine. But a 1-to-1 ratio happens to be one of the degenerate cases where it actually makes things worse. And so even if you had like 25-75, then you could easily identify V4. But if you happen to have it 50-50, then you still end up with problems. But I don't know if we'll get to that, to explaining why these 50-50 combinations don't work. But what I can show you is why certain tracers don't work and why other tracers do work, and how to choose between them. And so that's going to be the next section right here, studying the effect of good and poor tracers. So normally, the process for calculating the steady state fluxes given the measured MIDs is as follows. You guess a plausible flux distribution. It satisfies FBA, for example, but MIDs give you more constraint information. So then you solve for the MIDs that this flux distribution entails, and then you compare the measured MIDs with the calculated MIDs. And if they're close enough, then you return the flux distribution. You say, oh, that flux seems to have produced the MIDs that we actually observe. But if it doesn't, then you have to go back to step one and guess another flux distribution, and hopefully one that's closer to the real flux distribution, the one you chose previously. And fortunately, our network is small enough that we can actually calculate an analytical solution. And so we'll use this solution to study the effect of good and bad tracers on our ability to calculate steady state fluxes. So if you recall, beforehand, I said that not only can we decompose our EMUs into separate networks of different sizes, but we can also combine them into one network as well. And so this is what the combined EMU network looks like. Okay, so again, we have these three tracers coming in. So we know what A123, A3, and A12 look like. We know their MIDs because they were given to us. And what we want to calculate is the flux for V4, which is, so the flux for V4 given the MID of E123. Okay. So each EMU, so we're going to introduce a new concept. We've discussed EMUs, EMU reactions, and EMU networks. Now we're going to talk about two new concepts. One of them is called an EMU basis vector, and then one is an EMU pathway. So let's talk about EMU basis vectors first. Each EMU basis vector can be viewed as a unique way for assembling substrate EMUs to form the product. So by definition, so when I say substrate EMUs, I'm talking about those original the original isotopic label that we gave it. So and we want to think about the product, which is in this case E123, in terms of A123. So by definition, EMU basis vectors have the same EMU size as the product. Importantly, given a network and a labeled substrate, one can associate an MID with each EMU basis vector. Ultimately, one can interpret the MID of the product as a linear combination of EMU basis vector MIDs, where the coefficients of that linear combination are solely a function of free fluxes. So the coefficients quantify the fractional contribution that each EMU basis vector contributes to the product. Thus, by definition, the sum of the coefficients must always equal 1. So for the simple model, an analytic solution exists that can be written in terms of the EMU basis vectors and the free fluxes. So here's E123, and we can take a look at the EMU for A123 and A12 convolved with A3, and this MID here is a linear combination of these two EMUs. Does that make sense? So I don't have my Discord open. Let me pull that open. Okay, here we go. I can see if people are giving us a thumbs up. So that's a good sign. Okay. All right. So now, so this is the analytic solution. So if I knew what my V1 and my V4 was, then I could calculate it. But even if I don't, if I know my E123, I can back calculate to figure out what V1 and V4 must have been. And so that's what we're going to do here. So here's the EMU basis vector for E123. It's basically this product of A123 and A12 convolved with A3. And now we're going to talk about EMU pathways. So in addition to the EMU basis vector, they also define an EMU pathway. Each EMU basis vector specifies a unique EMU pathway from substrate or substrates to product. And so actually this is very closely related to elementary modes. And and so if you recall the elementary modes previously, these were networks of minimum cardinality sets of reactions that form a steady state. And I'm going to assert here that the elementary modes of the EMU network are in fact the EMU pathways. So here are the EMU pathways. So I used my elementary modes analysis to calculate the EMU pathways. So the first EMU pathway is A123, and that's what this looks like. And the second EMU basis vector, the second EMU pathway is A12 convolved with A3, and that's what this pathway looks like. And so what we're doing is we're saying, okay, this MID here is the result of combining A123 with this other pathway, this A3 convolved with A12, to form A123. So let's see what happens when we send different tracer combinations through that EMU network. Okay, do we have any questions so far?
Not at the moment. There was a question about the ratio. I answered that we used 1-to-1 in the first example to show how we can calculate the MIDs.
Okay, so let's see. Set 1-to-1 ratio. Or we should supply them in 1-to-1 ratio. Yeah, you put several times 0.5. Oh, right. So I did show them as 50-50. And the reason for doing so is to actually show what happens when what goes wrong when you actually do make them 50-50, right? So in this case, in this small model, it doesn't matter. But when we just slightly extend the model, it turns out to matter. And if we get that far, I'll show it. Otherwise, I'll just mention it at the very end. So here we have 4 different tracer compositions, right? The first one is 100% labeled, 100% one labeled. Second one is 100% fully labeled. The third tracer composition is 50% 1-3 labeled and 50% 1-2-3 labeled. And the last tracer composition is 50% one labeled and 50% fully labeled. Now, what's interesting is that because we have discovered these EMU pathways or EMU basis vectors, we can calculate the MIDs of each of those basis vectors based on the tracer composition, right? So in this case, the MID of A123 is just going to be 0, 1, 0, 0. And likewise, when we calculate the EMU of E12 convolved with A3, we end up getting A10, 0, 1, 0, 0 as well. So this is a problem, right? Because I cannot use the MID to distinguish whether or not the flux came from A123 or whether it came from A12 convolved with A3. And likewise, when I send in a pure tracer of fully labeled, I get the same problem. The MID from A123 and the MID of A12 convolved with 3 is the same. Okay, so we split them up into separate tracers. So now I have 50% 1,3 labeled and 50% 1,2,3 labeled. But when I calculate the MID, I get something that's different. I get 0, 0, 0.5.5. But when I convolve MID, when I convolve A12 with A3, again, I get 0, 0.5.5. It's only when I use the 50% 1 labeled and 50% 1, 2, 3 labeled, fully labeled, that the MID for A123 looks different from the MID for A12 convolved with A3. It's in that case specifically that I can identify a unique value for V4 that from those two tracer compositions. So another way of saying that, so this is the exact same graph, just shown in a slightly different way. Here's my EMU basis vectors. Here are the EMUs associated with each of those basis vectors. A123 is just by itself, but A12 convolved with A3, I can separate those EMUs out by themselves and show which ones are. I got some indentation issues here, but then I can calculate their MIDs. So that's exactly what they did up there. And then I can plot them. So when I plot the MIDs, what we see here is that only in the case where we have 50% 1 labeled and 50% fully labeled, do the MIDs for each of those EMU basis vectors look sufficiently different that I could back calculate what flux distribution must have caused one pathway versus the other pathway. Does that make sense? Yes, yes, yes. Okay, so we got at least 2 people who understand what's going on. That's great. Okay, now I just want to make sure that people understand that what you see is not the red and the blue. What you end up seeing is just the observed MID is what happens when you add those two together. And so the observed MID will look like this. So it's only in this case right here where we have 1 labeled and fully labeled that you have an MID. And then we can then say, all right, well, because we know that M+2 is only came from A12 convolved with A3, that we can now separate out the 2 branches of the pathways. So just to help you guys remember, oh, that's not the, I need the combined one here. Emu. Go ahead and pull that up. Combined. There it is. All right, so here's the EMU combined. And it's only in this case that we know the A12 convolved with A3 was responsible for this M+2, guy. And we know that from here. Okay. So how are people doing? We're at 125 right now. And I think the only thing I want to do now is just kind of address the question about, well, why not 50%, 50%, right? Because it wasn't clear from this, because I could still identify all the queries. The point is that if we extend the model, so here's our original model, right, going back to E, but if we now extend it so that C1 can escape and D can combine with B to form E and G, two units of G, and then we allow E to go to F, then and we now have all these different so now we have a lot more, we have much more rich elementary basis vector, EMU basis vectors and EMU pathways. I'm not going to go through it too much. I'll just show you what the EMU, so this is the network itself, the elementary modes of the network. And then here's the EMU size 1 network. Here's the reaction network. You can, all these calculations are the same, so you can calculate the reaction network. You can calculate the balance reactions. So you can solve for the MIDs that are produced by the flux distribution that we showed above. But importantly, and likewise for EM2 size 3. But importantly, if I can get down there, so these are all the EMU pathways now from the combined network. And these are the tracer compositions. So here we go. So what they're showing here is that if the EMU basis vectors overlap with each other, that means that you've lost a rank. So there are rank 3. So there are 3 EMU basis vectors in this slightly larger model. And if one of them overlaps, then you end up getting rank 2. And if both of them overlap, you end up getting rank 1. So if all 3 overlap, then you end up with a rank 1. And what they show is every time you, so the black ones means that, so every time you have a black one, that means that it's full rank, but there can still be cases where they're observable except for 50-50 mixtures. So if you chose anything other than a 50-50 mixture, then you would be okay. But because you chose 50-50, then you cannot distinguish one branch from another branch. It just works out math-wise. They don't. How many metabolites should be traced and quantified to be able to determine the relative fluxes of multi-step, multi-branch pathways? Is determining the substrate and final end product or do we need to determine also the metabolites of each branching point? That is an empirical question. It really depends on the network itself and what you're able to measure, because oftentimes you don't have a choice. You know, the set of metabolites that you can measure is limited by kind of what's observed, you know, what can be, as you learn from the metabolomics day, that not all metabolites can be measured in all conditions, right? Sometimes a metabolite can be measured on one condition, it can't be measured under another condition. And so it really depends. So what I do recommend is that before you do 13-CMFA, which is very expensive, by the way, these labeled metabolites are not cheap, is that you perform a metabolomics experiment on the unlabeled data, on the unlabeled, you perform an unlabeled metabolomics experiment to see which metabolites are even observable at these levels. So you set up the experimental conditions exactly the way you would as if you were going to produce, if you were going to do a labeled experiment, but you just don't include the carbon label, and you see which metabolites are observable. And those are the metabolites that you'll be able to get an MID for. And so given knowledge of which metabolites you can get an MID for, then you can determine which fluxes you can actually observe. Did that answer your question, Ahmed? Oh, yes, clear. Thank you. Okay, cool. And the other folks who have questions, we're at 1:30 right now. And I know we've got some tutorials that we're going to break for, but I'm happy to answer questions. Yeah, there we go. So it takes a couple of days to post, but the video will be available. Have a terrific day, evening, everyone. Go ahead and post your questions in Discord or chat. I'll be happy to answer as I can.
Thanks, Jeremy. Quick, before we move on to the next section, I want to thank everyone who joined us today to the open session. Now's the time when we're going to flip over and spend some one-on-one time with the students, our 25 students. So I do thank everyone else who joined us. One of our communications team is actually going to send out a survey later, and we're interested in your feedback. We've been doing these summer schools for a few years, and last year we started this virtual part where we open it up since we're all at home anyway. And so we're interested in your thoughts on what's useful, what isn't as useful, and how we could grow and continue to make this summer school better and better. So we will ask for your feedback, and if you have anything, please do send it. We are interested in that feedback. And we thank you again for attending. This was a fun week for us, and we hope that you all enjoyed it and learned something. So for the students, we'll flip over as we've done every day. We'll switch to the other Zoom where you can all interact more closely with the instructors.